study in Stanford University, in particular the PhD thesis you did there. And then you've been a postdoc in statistics at Columbia University and the Simmons Foundation Junior Fellow. And then more recently, you've become a Chief Scientific Officer for Responsive AI, and you're also Assistant Professor of Neuroscience in Psychiatry in Cornell University. So I would say that your career has been dedicated to psychiatry, but with a strong methodological flavor. So I think you have a lot of things to say to many of us at Neurospin, and that's great. And so this morning, you're talking about biological subtyping in psychiatry, hope, controversy, and a new method.
That's right. All right. Thank you. Thank you very much, Bertrand. Thank you for having me. It's great to be here at Neurospin. And I hope it sounds like you've all had several months to rest. I'm going to go through a number of, can we hear me? Can we see me? There we go. All right. Sound? Okay, nothing?
No.
Should I just be very loud? Right, we're gonna do. How's that working? Good, good, great. All right. So I recently, during the pandemic, in the end of 2020, started a lab at Cornell, and we're focused on really neuroengineering and neuropsychiatry and computational psychiatry. So really neuroengineering applications in the psychiatric domain that span from individual human neurons through spatial distributions and spatial transcriptomics, all the way through developing new models for that type of data. And today, I'm going to be talking primarily about just human neuroimaging data. So I think more relevant to your interests here, as well as some new statistical methods for biological subtyping. And in biological subtyping in particular, what I'm referring to is using neurophysiological measurements such as neuroimaging data along with clinical measures to come up with a better biological categorization of patients in particular. And I'm going to go through kind of three parts here. The first, why we hope this might work, why we think that biology can help guide better psychiatric diagnoses, and by that we mean, of course, improve them so that they are relevant in particular to treatment and to understanding disease etiology, more so than they are now. So I'll start with two examples of why we think that's the case. Beginning really with what the problem is, which is that historically psychiatric diagnoses have been troublingly heterogeneous. And this is exemplified by our current common best method for psychiatric diagnosis, the Diagnostic and Statistical Manual of Mental Disorders. Here's the latest version, the DSM-5. And just as one example, if you are diagnosed using this manual as depressed, it means that you've qualified by having 5 out of 9 items on a checklist. So there are literally hundreds of different ways, different combinations of mood, anxiety, behavior that can lead you to be categorized as depressed. And when you talk to clinicians, this is very clear. Some of their patients are bouncing off walls with anxiety. Some of them can't get out of bed with inertia. They both go into the same bin, despite what appears to be very different underlying biology. And we think that really lies at the heart of some of the problems with treatment and treatment responses being unreliable for psychiatric disease generally. So a few years ago, myself and my colleague Connor Liston were thinking about this and formulated a very simple hypothesis. I don't think many here would disagree with it, that distinct neurophysiology is at the root of heterogeneity in psychiatric disorders. And so the idea here is really, the reason this is contrary to sort of historical approaches is that rather than starting with symptomatology, we're going to start with the neurophysiology and let that guide us in terms of let us cluster patients into relevant categories rather than the other way around. So all pretty straightforward at this point. What are we going to use as our measure of biology or of neurophysiology? something, again, very commonly used, broadly used, that I'm sure you're all familiar with. We're going to use resting state functional connectivity as our measure of neurophysiology. We're going to do this by using a parcellation, in this case, the Power Atlas, which is a very longstanding, well-validated parcellation of 258 different spherical ROIs across the brain, cortically and subcortically. And for each of these, as I'm sure you're familiar, we can just very simply derive a correlation over time of the bold signal, averaged within each of these ROIs. For example, here, if we look at the dorsolateral prefrontal cortex and the posterior parietal cortex, there is a correlation of about 0.88 over time. So we're not even doing anything with dynamic functional connectivity here. This is all very established in the literature. But even with this relatively simple method, we end up with, if we look at the upper diagonal of this correlation matrix for one patient that's been organized here by different well-known regions and networks, we end up with on the order of 30,000 different correlations. And we can see that even in this one individual, there's quite a bit of structure. Some of that structure may bear on psychiatric conditions we're interested in, some may not. There's a lot of variability that simply may not interest us. And so when we started discussing this and the problems inherent in it, there are two big ones. The first is, what do we do with so many correlations when inevitably we have at best hundreds of subjects and we have tens of thousands of variables? So there's a variable selection problem and a multivariate problem. And then two, how do we parse out the variance that's really relevant to what we care about? And to do that, we turn to what we'll call multi-view statistical learning. I'm going to give a very simple example, use the simplest possible example of multi-view machine learning, which is canonical correlation analysis, motivated by this idea that there is some true underlying brain state, and we're getting kind of different noisy projections or different noisy measurements, partial measurements of the kind of true underlying biology that measures. So this is very cartoonishly depicted here, or we can show this very simple factor graph, defining this model in which there's some latent state that we think is lower dimensional Z, and from that we're getting clinical measures and neuroimaging measures. Again, this is a very classic statistical method dating back to Harold Papelling in the 1930s, and really is just trying to look for coefficients A for the clinical measures and coefficients B for the neuroimaging measures that are going to maximize the resulting correlation between applying those coefficients to our clinical measure, or multivariate clinical measures X and multivariate neuroimaging measures Y. So it's deceptively straightforward, but turns out to be quite effective, in part because it's going to allow us to do some filtering of the neuroimaging data using what is relevant to the clinical measures. Again, this brings us back to this sort of structure and looking for which distinct neurophysiology might be the root. Initially, we began with treatment-resistant major depressive disorder, MDD. And this was published in this work in 2017. Rather than the DSM-5, we're going to use a very well-validated scale, the Hamilton, the HAM-D, or the Hamilton rating scale for depression, which is sort of the gold standard 17-item scale for assessing depression across different symptoms associated with it. For example, anhedonia, anxiety, agitation, sleep disorders, etc. And as I said before, as our neurophysiology measure, we're just going to use functional connectivity. And this is going to be done across a multi-site study collected in several hundred individuals a while ago, at this point, several years ago. So if we look at 220 of these individuals and we run first in feature selection, so this is just looking at the Spearman correlations, filtering based off of Spearman correlations, essentially across the functional connectivity measures that relate strongly to the these 17 items in the HAM-D, and then run our canonical correlation analysis. We find that there are two very dominant latent dimensions. The first, if we look at the coefficients related to the symptoms described in particular by anhedonia, a little bit by psychomotor retardation, and the second defined in particular by anxiety with some loadings on insomnia. This is already compelling. Note that these are fit to the training data, so this is not this looks like a very tight correlation, but it is inevitably overfit since it's on the training data. However, we found this robust lower dimensional space, and in that lower dimensional space, we applied hierarchical clustering. Again, another very straightforward classical method. It's been around for ages and ages. We applied very standard methods, several of them for finding the number of clusters. They agreed all on there being 4 clusters in this data. We'll come back to whether or not we really think these are separated clusters or not later. But for now, you can see that along these 2 dimensions, the anxiety-related and the anhedonia-related dimensions, clusters 1 and 3 in particular seem very anhedonia-related, or in particular had their variance along that dimension. And the anxiety-related connectivity was split kind of more across 2 and 4, we'll say. This was important because each of these dots is a subject, and each of these subjects also received repetitive transcranial magnetic stimulation in their dorsomedial prefrontal cortex as treatment for treatment-resistant MDD. And we saw the strong, significant differential response in the groups. And that mapped in particular onto these clusters 1 and clusters 3 along this anhedonia-related connectivity dimension. This replicated in a prospective group of 30 subjects collected subsequently, in which we built classifiers on this data and showed that we could predict who would and would not respond. And so this kind of gave us this first belief, this first hope, that using these relatively straightforward methods, essentially using CCA or using a multi-view machine learning method to filter with neuroimaging measures and clinical measures, and then clustering this low-dimensional space that results, could give us some purchase on predicting treatment response and getting at diagnostic categories that were relevant to treatment assignment. So this is the first study. Just to sort of recap, we can identify 2 brain behavior dimensions that predict individual differences in anhedonia and anxiety-related symptoms. We can do hierarchical clustering, again, very classical, to reveal 4 putative subtypes of depression. The subtypes are associated with different clinical symptom profiles and critically with different treatment responses to repetitive transcranial magnetic stimulation. And again, these are treatment-resistant patients, so they've already failed 3 cycles of treatment. And we currently have large-scale clinical trials underway in several hundred patients to test the efficacy of these biomarkers for predicting TMS responses. Those are looking quite promising. And so this was the work that first provided some indication to us that perhaps this approach, or one like it, could work in a robust, meaningful way to start with biology and work our way toward relevant diagnostic categories. Okay. But can this generalize beyond depression, and can we sort of improve these methods? That was sort of just our first attempt. So we turned to, again, some more publicly available data sets for heterogeneous, in this case, a heterogeneous neurodevelopmental disorder, autism. Autism is classically heterogeneous. It's sort of defied categorization across every version of the DSM, and gets grouped, regrouped, or split, or amalgamated. It is, however, known to have a strong heritable component, about 85% heritability. So we believe there to be real biology underlying it, but it's been very difficult to tie that biology to categories that sort of make sense within autism. So again, we started with this very simple diagram and thought, how can we use distinct neurophysiology in the form of resting state functional connectivity in publicly available data sets such as the ABIDE 1 and 2 and R to get at the root of heterogeneity in autism spectrum disorder. In this case, we're now going to use the ADOS-2 since that is what is available to us. In this case, there's only 3 measures: social affect, restricted and repetitive behaviors, and verbal IQ. And again, we're using resting state functional connectivity from a multi-site publicly available data set, ABIDE, which many of you may be familiar with. There's several hundred individuals that we end up with after properly accounting for motion, which is worse in this cohort because they're children. We end up with about 300 individuals, and we're going to put them through the same analysis, but now improved a little bit, really to account for this issue that in solving this problem, you can imagine this is really just solving a SVD, or if you center a PCA, where on the cross-covariance matrix between your clinical measures and neuroimaging, that's the sigma xy. But before doing that, as part of the method, you have to whiten on the left and the right, which involves inverting these matrices that are the correlation matrices or the sample correlation matrices for, for both those multivariate data sets. That's very unstable when those are very correlated. So a very simple thing you can do, again, this is not a new method either. It dates back to the 1970s. We just found it to be very effective in this particular application, is to add regularization to the sample estimates of the covariance matrices for your clinical measures and neuroimaging data. This gives you two hyperparameters you need to tune. To do that, along with the feature selection, we add additional layers of cross-validation. This looks rather complicated, in part because it is, but really at the heart of it, things are pretty simple. There's just a nested cross-validation inside of a larger cross-validation to get an unbiased estimate of the test canonical variates. Again, although it's conceptually simple, I've been trying to encourage people, just as a result of recent publications we'll revisit shortly, to include diagrams like this in their papers, so that you can see exactly what's going on at each step. Here we're doing some bootstrapped feature selection. So this is stabilizing our feature selection within this inner loop. We're tuning our hyperparameters within this inner loop, and then we are on an outer loop, testing on some held up data that has not been seen by any of those previous stages. We're doing that 1,000 times, and we're looking at the modal results. And this is all, the details are in this Booch et al. 2023 paper. So doing this, excuse my animations, gives us 3 significant latent dimensions. Here significance is established against an empirical null distribution that we built through shuffle tests. And the dark dots here are the training data, and the lighter dots are the test data. You can see that there is, as expected, some overfitting on the training data, but there's still some significance. The statistics in each quarter are for the test data. They're strong, reasonably strong effect sizes. They're significant. And we see that they map onto, if we look at the coefficients associated with the symptoms, the first primarily onto verbal IQ, the second primarily onto social affect, and the third on verbal IQ, sort of a mix of verbal IQ and in RRBs. Again, doing just what we did before with depression, we now use hierarchical clustering in this latent space. We again, using 6 different measures, all of which agree on this, come up with 4 clusters now in a 3-dimensional latent space. Here's an example of one, the Klinsky-Hrbach. And doing this over 1,000 replicates, subsampling, yields sort of the same stable clusters and the same number of clusters. So that's reassuring. Looking at the resulting subtypes, if we consider those 4 clusters to be subgroups or subtypes of autism, we see that there's some interesting differences in their clinical symptoms. In particular, the group 4 is quite spared in terms of social affect. Group 3 is very spared in terms of restricted and repetitive behaviors. And then there's this interesting split on verbal IQ between groups 1 and 2. Here, higher is worse for social affects, worse for RRBs, and better for verbal IQ. So this group 1 has relatively spared verbal IQ, for example, as well as differences in total severity. So group 4 is relatively spared in terms of total severity in addition to social affect. So this is interesting. And again, I'm not going to go, there's a lot we can potentially say or discuss about the functional connectivity related to these subgroups. It does relate to findings in the literature. It is most of these, just basically looking up here, most of these groups, most of these areas and networks are ones that you would anticipate finding based off of previously published literature. So we have the limbic, the MN, frontoparietal, salience network, of course, sensory, cerebellum, based off of recent work seems reasonable. And there's interesting and stable different types of atypical connectivity patterns. This is measured against normative controls. You can think of these essentially as T-maps against the normative population of about 1,000 individuals run through the same protocols. And since my primary goal today is just to be just to convince you that you can believe in biological subtypes. We'll neglect a lot of the interpretation here, but there's a lot of it in the paper. Critically, if we run the exact same analysis pipeline that we ran on the ABI data, which is what I've been showing you so far in these 299 subjects, if we run that in a completely different data set collected by different scientists and different scanners, and in parallel with no overlap between the analyses, we recapitulate very, very similar results. This was quite surprising to me. I anticipated these to be more different than they ended up being. But we see both in terms of the clinical symptomology and in terms of the connectivity, broadly very similar patterns. Again, more can be done with this, but I'm just trying to convince you here that these are robust. Another thing we'd expect if these are real is that if we look at those patterns of atypical connectivity, we might be able to relate them to other biology that would be consistent with a diagnosis of autism. Again, knowing that this is 85% heritable, we decided to regress on spatial transcriptomics across the brain. So as a result of the Allen Human Brain Atlas project, there's very beautiful data sets for now 6 individuals. We can look at their spatial transcriptome across the brain. We can bin it into the 258 ROIs from the Power Atlas that we use to define our functional connectivity. And then for each of those atypical connectivity matrices that we've estimated per subgroup, we can run a partial least squares model. So you can think of this as a large dimensionality reducing regression. It's actually very closely related to CCA as well. And we can use standard conservative tests for significance on the genes. Here we're using both a spin test and then another test of permutation to decide if any of these genes and the distribution of these genes across the brain is significantly related to these atypical functional connectivity, and is that different across subgroups. Perhaps most importantly, to just validating our idea that these biological subtypes have teeth, is that if we look at ASD gene sets that have been published in the literature from SFARI and SPARC and other initiatives and recent work, we find that genes known to be downregulated or thought to be downregulated in autism, and those thought to be upregulated have across these different subgroups exactly the direction coefficients that you would anticipate. So this is very simple. It for us was initially a sanity check, but it's important to, again, establishing that there's real biology underlying these subtypes. We also would like to see that in non-psychiatric gene sets, such as heart disease and dementia, we're not seeing correlations, although ones that are related to autoimmune disorders in the CNS and arthritis, both with autoimmune components known to relate to autism, do show significant positive coefficients. Interestingly, if we look at other neuropsychiatric gene sets, some of which are thought to have correlation with autism, most obviously vocal learning, but also schizophrenia and ADHD, we see that there are significant correlations, in this case negative, but they are also not homogeneous across these different subtypes, and that is also true for synaptic signaling gene sets. So in particular, for example, in subgroup 1, we're seeing what seemed to be a large and significant negative relationship, very different from group 2. Again, we go on from here in the paper, if you're interested in the details, to look at these in terms of protein-protein interaction networks, and further kind of tease apart what might be going on in the different subtypes and how that relates to their atypical connectivity. I'm not going to do that today. Please see the paper. What I, again, would like you to just take away from this is there is this, what appears to be a strong, significant correlation between known biology and autism, and these subtypes we're able to find by using relatively standard statistical methods to relate clinical scales to neuroimaging data. And we can show this. We, again, have run the controls you'd sort of expect to establish further that these appear to be real to us. So again, they generalize across data sets. They're stable across clustering methods. We applied 6 different clustering methods and different measures of distance in those clustering methods. They're stable across age. So in the ABIDE data set, there's actually a pretty broad age range. So we restricted and reran our analysis on a smaller range. And also used a mixed effects model to establish the impact of age and did not find one. They're stable across parcellation. So in addition to the Power Atlas, we used the Crowd 200 Atlas. They were stable across how we were, where we were getting our gene expression atlases from. So we recapitulate our results across the brain span instead of the human brain atlas. And we also finally, to convince ourselves, in particular, far down this analysis pipeline where we'd gotten to protein-protein interaction, that we were fine that these results were interpretable in a way that related to literature. So we ran this independent text mining analysis, which I think is somewhat interesting, where after identifying, see in the center here, after identifying the protein-protein interaction hub genes, and taking the up to 10 most connected genes, we could convert those to entry or mesh IDs, go to PubMed, download abstracts that included those hub genes, and then based off of a set of reasonable keywords that are in the appendix, related to each of the different clinical measures of interest, we could calculate the relative frequency of each subgroup's hub genes, and how associated they were with these behavioral keywords for, again, social affect, restricted and repetitive behaviors, and verbal IQ. And just to remind you what those look like in the different groups, I'm showing you this previous result. And what we found is, once again, group 3 was consistently very spared in terms of RRB. Group 4, very spared in terms of social affect. And again, a sort of more consistent relationship between language words for 1 and 2. So this again is all meant to try and convince you and us that at the end of these statistical analyses, there are real biobehavioral effects of interest, and they can be captured and quantified robustly across data sets, and they generalize out of samples. Okay. So we were pretty happy with this, but not everyone was. So around the time we published Drysdale, this came out in bioRxiv from the Markman lab, Dinga et al., evaluating the evidence for biotypes of depression, attempted replication of Drysdale et al., 2017. It was subsequently published in Neuroimaging Clinical, and the overall finding was they argue that the methods used in Drysdale et al., 2017 do not provide convincing evidence for biotypes of depression. So at this point, we thought they might be directing this at us. We weren't sure, but we became concerned and went back and evaluated why their results may have been different from ours. And this was we find this very instructive. So, and it's a good lesson, I think, for those of us who do statistics when we pick up these data sets and start doing analysis. It's very important to understand the particulars of the domain. And so, again, the sample size was similar. They had fewer per scanner. They had more scanners than we did. But in our case, it was very important to us and our inclusion criteria that all subjects were having an active episode of depression. They were not in remission, and so currently active and meeting criteria for treatment resistance. And they all had moderate to severe total symptom scores. Their subjects were psychiatric outpatients who did not have to actively be having an episode of depression, and at DSM-4 for either MDD, panic disorder, social phobia, or generalized anxiety disorder, with again, no specific requirements for active symptom severity. This is a very significant difference in cohort, and it's critical if you're looking for subgroups that might be in the tens to narrow your scope to actively sick individuals if you already think that's a heterogeneous group. So we think that's really the key take home here. There were also differences in their motion artifact correction and some of the other controls. But to convince ourselves further, we took the same data sets and we ran it. What you're seeing in the histogram here is just the Z values associated with the correlations between all of the 17 items in the DSM, sorry, in the HAM-D, and all of the available functional connectivity measures. And we use, since they're inevitably going to be correlated, we use this correction from Bradley Efron, initially developed for genomics, but that allows you to take into account the correlation between the different tests you're doing, and come up with, at the end, the sort of significant Z values above expected threshold. And in this case, you can see that in the data set that didn't get all used, very few of these wander north of about 1 in terms of Z value for the data set that we used in Drysdale et al. A good number of these were quite significant. Again, I think this mainly speaks to the fact that our data set was very, very carefully selected and very well cleaned to have direct bearing and relevance on treatment-resistant depressed patients. In addition, there's now growing evidence literature for biological subtypes. So this is also reassuring. A number of papers have been published. We recently, in the lower corner here, Katie Dunlop has published a very nice paper on the 3D data set from Montreal. So an independent MDD, treatment-resistant MDD study showing that the results from Drysdale et al. Do replicate. But all of those papers that I just cited that would make us think that perhaps this method worked, all leverage data sets on the order of hundreds of subjects. So another major issue that arose recently in the literature that I'm sure you are probably aware of, this paper from Merrick et al., which claims that reproducible brain-wide association studies require thousands of individuals. In particular, they're talking about studies of the associations between inter-individual variability in human brain function and cognitive behaviors or psychiatric symptomology. So this is very broad. This covers pretty much most cross-sectional fMRI work in these large open data sets that have been recently released. And their claim is that any brain-wide fMRI investigation studying correlations between brain and behavior across a population should require thousands of individuals to be reproducible. Why might this be the case? They show it for both univariate, what they're calling BWAS, so these brain-wide association studies. Again, just cross-sectional fMRI in populations. The univariate correlations are incredibly small across the Human Connected Project, ABCD dataset, and UK Biobank dataset. These are in thousands of individuals. And they're looking at correlations here between, again, functional connectivity and as we work, and either the NCI toolbox, so cognitive behavior, which is known to have strong effects, or psychopathology. And here on the right, more damningly for us, they're looking at multivariate BWAS. So in this case, looking at the psychopathology measures in the ABCD toolbox, and showing the way to read this plot is because they had such a large data set in ABCD, they were able to split it in halves. So they could split it in 2,000 individuals. They could hold apart 2,000 of the individuals and run their analysis in the initial set. And they could change the sample size in that set from 25 through, in this case, 1964. The color of each dot here is each dot is a different run. So for example, for one dot that's deep purple, they selected 25 subjects. They calculated the in-sample correlation R that they're showing on the y-axis using either a support vector regression or, as we do, a canonical correlation analysis. They come up with an R that's pretty high, say 0.8. Then in that completely held out sample, they take that same model, they apply it, and they find that the R is actually close to 0. So even in the multivariate case, where we would hope that we'd be much better powered than we are in the univariate case, they're finding that we're in trouble. Again, if true, these are a major threat. Why would you fund fMRI research, which costs around $1,000 per individual for these effect sizes? They're showing, in particular, the univariate Rs that are median 0.02. And why should we believe past brain-wide association study results like ours or those involved in biological subtyping? if thousands of individuals are necessary for reproducibility and they only have hundreds or tens. It seems more likely that those results would be due to selection bias in publication than they would be due to real results based off of what they're presenting here. And this was not lost on the media. So perhaps most importantly for funding, most major news networks picked this up and trumpeted the same result. However, there was a really critical oversight in the way that they did their analysis. So it was a pretty straightforward analysis. They started with a principal components analysis to do dimensionality reduction, which ensures stability, and kept about 50% of principal components. And then they fit a support vector regression to their sort of discovery sample, and then compared that to this out of sample replication study. So we can just show this like this. On that y-axis, their results are fit in this training sample. They get that model, they report the R, then they take their model and they apply it to this held out sample. They get an R there. They plot those two against one another. In a very nice Mattis arising, in reference to this paper, Spizak, Bingle, and Weiger show that if you just do standard cross-validation within the training sample, which is what you would do if you're trained in machine learning, generally speaking, if you want a good estimate of what your holdout sample result is going to be, you get very different results. And again, they do exactly the same thing, but now they use training validation, test cross-validation on the discovery sample. So again, this is probably all very straightforward. You're already aware of it if you're familiar with machine learning, but it's a big deal because there's a lot of people applying machine learning now to these methods, and you can come up with pretty wild conclusions that might grossly divert funding away from your area if you're not careful about how you do your cross-validation. So in this case, this is the correct way to do it. You would do training. If you have any hyperparameters, fit them on your validation set. If you're doing any feature selection, also do that there. Then get your test on this holdout sample, and that would go on your y-axis. And then out of sample testing would be in that out of sample set that never saw any of this. So the Weiger, the Spizak, Bingle, and Weiger paper shows this on the HCP dataset, which I think they already had in hand. Because of the importance to our work and because of the American Turbo-Clemens, the ABC dataset, we also, in our hands, reproduced essentially what Spizak, Bingle, and Weiger had found, but in the ABCD dataset. Here, these results are for support vector regression. And what you can see is on the left of each of these, if you use the approach in Merrick et al., you see exactly what they saw. There's an overestimate because you are overfitting in your training data set before you estimate on your out-of-sample data set. And then if you correct things by using cross-validation in training, you see out-of-sample, again, you see this, essentially what you want is for these dots, as your sample size on average increases. So for example, this yellow, orange, red dots, those are each the average for the other dots corresponding to 100 studies, 100 different samples, different training set sizes. You find that first, they lie along a nice x equals y line, such that your in-sample estimate is correctly predicting your out-of-sample estimate. Perhaps more importantly, for the current discussion, these are significant in the tens. So the dotted line here is significance against an empirical null distribution generated exactly as suggested in the American Turbo-Clemens. And we see that tens of samples, while there's a lot of variability in the outcome, as shown by the spread in the dots, is actually north of what would be considered statistically replicable. That's very different than thousands, clearly. We can do the same thing for, again, this is for cognitive ability using the NIH toolbox. We can do the same thing using RCCA, which is, again, important to our biological subtyping results. And we can do this for cognitive ability and for psychopathology. For cognitive ability, which is known to have stronger relationships with functional connectivity, again, tens of subjects seem reasonable. For psychopathology, in what is a very mixed sample, including a lot of normative individuals who are not actively in a psychiatric episode, we find that about 100 is sufficient. And we can quantify this more clearly if we look at training set size on the x-axis and we look at the percent of significant out-of-sample results across 100 different replicates of this analysis. we can find that both for support vector regression and RCCA on the cognitive behavior, you need very few subjects. You're getting before 200, you are saturating on your reproducibility. And for support vector regression and RCCA on the psychopathology data set, it may require up to 400 or 500. In this, again, very mixed population that has not been selected for clustering on psychopathology. So in some sense, more similar to the Marquand, the Digga et al. Data. And still, you see strong replicability and significance. If we look at effect size, so that's the actual autosample R, we see that we recapitulate the same sort of saturation curve. So again, the data sets, the data set sizes they use in the paper are on a log scale. If we plot it on a standard scale, as the small plots on the bottom show, then for all 4 data sets, there's this sort of logarithmic asymptote in effect size, and the same is true of the coefficients. We're working on, there are some nice explanations for why this is the case using random matrix theory that we will have out relatively soon. But the most important thing is not only do we not require thousands of individuals for reproducibility, these studies don't benefit from it. So you're actually potentially throwing away government money if you're collecting more and more subjects for one of these studies. So some quick conclusions. Reproducible brain-wide association studies do not require or benefit from thousands of individuals, but they do require careful use of cross-validation. Tens to hundreds of subjects tend to yield reproducible, significant results consistent with previous empirical findings. That's not to say that you should be believing papers that have n's of 20. It's just to say that reproducibility is possible for strong effects at that level and may require tens to hundreds of individuals for less strong effect sizes. For multivariate analysis, there are diminishing returns past tens to hundreds of individuals in these data sets. And going back to the Dingadal work, cohort is critical. So diagnostic status and, of course, side effects and inclusion-exclusion criteria can have profound effects on your analysis. Further, in large consortia studies like the ones we were just discussing, the Mercadal paper, effect sizes for non-normative effects may be significantly diminished by inclusions of many normative individuals. So that sort of relates to both. So let me quickly go through. I'm approaching time here, but I want to show you, as a result of our analyses and then thinking through some of these critiques, we've also developed some new methodology. One of the most common questions we got about subtypes was, okay, if I look at that plot on the left, it looks kind of like it's actually maybe a continuous cloud. Like maybe there's just a dimensionality and you're imposing these clusters on that cloud somewhat artificially. These clusters look maybe somewhat more separated if we applied kernel density estimators in the autism work. But again, the same question holds: are these discrete? Are they continuous? And what does that mean for treatment? But there's problems really with trying to answer that question using a two-stage method like the method that we were using. Just to remind you, applying canonical correlation analysis and then clustering in this latent space that's defined by CCA. This problem has been known for a long time, since the 80s. We've recently published what we think is a good solution. And so just as a toy example, if we have some 3 ground truth clusters that have some sort of difference in means as well as their covariance, and we apply either, these are actually showing the first 2 variables of 200 variables. So this is just in a 200 dimensional space, and there are 100 observations. We see that spectral clustering followed by PCA does not do a good job of separating these clusters. PCA followed by spectral clustering does not do a good job of separating these clusters. But if we do our method, which is essentially jointly estimating both of those things together, we can actually get, again, in this toy data set, pretty good separation of these clusters. And the takeaway here is really just that what you want to do is jointly cluster and embed. And that makes sense because if you're applying a method like PCA that's looking for variance, and that variance doesn't happen to lie exactly along the dimension that separates your clusters, you're going to run into problems. This is sort of an analogy of Simpson's paradox, where you have maybe 2 different linear groups of clusters like this, but when you fit a line, it goes the opposite direction. And actually appears quite frequently in data that has covariant structure. So just as one more motivating toy example, again, this is sort of an XOR for clustering. If we have 100 observations in 200 dimensions of data like this, class 1 and class 2, and we apply PCA, it picks out the directions of maximal variance, which are these green and red lines for the first and second component. They do not align with the data. If we cluster, we get something like this, which is not a particularly good clustering. If we use our methods, there's 2 algorithms. I'm not going to go into the details today. I'll be discussing them tomorrow at the Henry Mind Group. But in essence, we're relaxing this parameter lambda in the corner, which allows us to go from one cluster, which is force all the data to be in one cluster. As it relaxes, the data is allowed to inhabit, in this case, 2-dimensional space of principal components that have to be both clustered and low rank. Another way of visualizing this, and another useful part of it, is that we get these coefficient paths out. So again, as we start with this parameter lambda very high, all of the estimates for the data are forced to lie in one cluster. That's what you see all the way on the left there, where it's about 134. And then as we relax it, the estimates for the data can relax into these 3 different clusters. And what you're seeing are the dark dots are the estimates of our methods, and the light dots in the background are the ground truth for these clusters. Of course, this is just toy data. I should mention this also can be relevant to the work I discussed before, extended to canonical correlation analysis. We're calling this pathwise clustered CCA or P3CA. There's a lot of details here, but fundamentally all that's happening is there's something like an alternating least squares step, but it's solving 2 convex clustering problems. Again, I'll discuss that more tomorrow. More importantly, if we apply this to public data sets, in particular public data sets that are relevant to us, where the number of observations, the number of samples and individuals we have is much less than the number, many fewer than the number of variables that we have. For example, the NCI SR BCT gene expression data sets, some more recently available mouse single cell RNA-seq data sets, some gene expression methylation data sets, and then just for larger data sets, these classic MNIST and MNIST image data sets, as well as one very large synthetic data set of 100,000 samples, and multi-view versions of those. We see that our methods, which here are in orange, PCMF, local linear version of that method, which is essentially the PCA version, or P3CA, which is the canonical correlation multi-view version, are outperforming, again, maybe most importantly, the methods we were previously using, such as CCA followed by clustering, or PCA followed by clustering, but also beating a lot of standard state-of-the-art methods. So elastic subspace, GMAD, Leibniz-Levin, Dirichlet process, Gaussian mixture models, convex clustering itself we are beating significantly, which is what our method is in some sense based on. So that's important, as well as 3 deep neural network methods. In fact, the one method that does tie ours, DEC, is a deep neural network clustering method, and that ties us just on 1 or 2 data sets. So we believe this is performative. It also works well on large data sets. Where you see X's or T's here, the X's mean that these methods couldn't even run on data sets of this scale, and the T's mean that they timed out at 24 hours, which was the cutoff that we set. Our method performs well here. Our method also can be applied to held out data. So you can check out a sample generalization. And again, it does well, we think, in these tumors data sets, as well as MNIST, fashion MNIST, and this large synthetic data. Coming back to the point, one of the points of doing this, besides just performance and working on data sets with not that many observations, if we wanted to find out whether or not we could build a method that allowed us to get around this 2-stage problem of projection into a lower dimensional linear Gaussian space, which essentially gives us this. So we just use the top 20 selected resting state functional connectivity values from the ABIDE data set from exactly the 299 subjects in Buch 2023 in Nature Neuroscience. And using CCA to embed and then clustering yields something similar to what we saw actually in the Drysdale, where you're not really convinced that maybe there's a boundary there. And what we suspected was that this is due to the fact that we're projecting on principal components, which are fundamentally looking for this sort of Gaussian linear space. Using our methodology applied to the same data, and again, relaxing that lambda parameter out from large and one cluster up to two clusters, we find that at least on these first top 20 selected resting state functional connectivities, we get very nice discrete clustering. And these particular groups, looking back and comparing them to the Nature Neuroscience paper, one of them in particular maps on well to the group with restricted and repetitive behaviors. Although we're doing more follow-up analysis now. And again, you get these paths for the coefficients that you can go back and interpret. So here the blue is social affect related, pink is RRBs, and yellow verbal IQ. And again, as you relax lambda, you see that you get groups of individuals. Each line is an individual, groups of individuals that split off according to these different their weights in these different variables of interest. So again, I'll talk about that more tomorrow. That's sort of a tour of the current work of my lab. Here's my wonderful lab. Most of this work was done by Dr. Buch, who's up on the upper left-hand corner, with some of the random matrix theory I briefly mentioned, done by Karsten Gimri, who's in the top center. And I just want to, again, highlight their names specifically. Thank Connor for being a fantastic collaborator. Thank my funding, and take any questions. Thank you.